pas même YouTube non plus.
Non. Pouh là.
Parce qu'elle est DG.
Ah là là. Est-ce que tu peux regarder sur YouTube si ça marche bien?
Alors, je veux bien, mais j'ai pas l'adresse.
Si, je l'ai envoyé à toute l'équipe par mail.
Ok. Ok. And it is working.
Ok, je te mets host du coup dans le Zoom. Donc, il va falloir que tu admettes Jean-Philippe Tyran qui se connectera.
JP.
Oui, Jean-Philippe Tyran.
Oui, mais c'est juste pour avoir le
Oui.
du prénom. Tu m'as regardé comme si je bien avec quoi. Est-ce qu'on peut faire quelque chose pour toi?
Non, merci.
T'as de l'eau dans ta gourde?
Oui. Je vais aller boire un coup. Ok.
Je suis 100%.
Ah bah attends, mais on veut pas de spoiler. Là,
Attends, je vais juste relire la première slide pour me lancer.
Et ça veut dire quoi ça?
Ça veut dire quoi?
Genre, P, il veut N, il veut
Corresponds to the requirement.
c'est plus des time series.
Mais il n'y a jamais eu de time series dans le film.
Non, mais à la base, c'est des time series.
Je vais les laisser rentrer. Mais il faut que j'aille boire. Et on va faire tous.
Est-ce que je lui dis bonjour et...
Oui, tu lui dis que les gens ne sont pas encore arrivés dans la salle. Je n'ai pas partagé une slide.
Ça veut dire quoi? C'est un peu rogue complètement.
Merde.
Hello Jean-Philippe.
Bonjour. Maëliss. Tu m'entends? Allô, allô.
Bonjour.
Oh. Comme vous parliez de moi sur YouTube, je me suis permis de me connecter. J'étais en train de vous regarder sur YouTube déjà.
Ah oui, bien sûr, on vient de démarrer, c'est parce qu'on était en train de mettre en place.
Ah ouais, mais comme vous parliez de moi, je me dis, bah, tant qu'à faire, je vais me connecter aussi, mais je ne vais pas vous embêter à un quart d'heure de démarrage.
Les autres membres du jury ne sont pas encore arrivés dans la salle.
Pas de souci.
Donc, on va attendre encore un petit peu. Et puis,
Je suis vraiment désolé, j'aurais aimé être là, mais ça, je me suis mal organisé.
Non, je comprends, c'est pas grave. J'étais surprise, honnêtement, qu'autant de gens puissent venir en
Ouais,
présentiel.
alors, je ne m'attendais pas à ce que Dimitri vienne.
Oui, moi non plus. Donc, c'était une bonne surprise. Donc,
Et
vous en faites
même
pas.
les Anglais, donc, enfin, les Gallois, pour être précis.
Oui.
Ça va? Je vous laisse vous concentrer.
Merci.
Je me reconnecte sur ce canal-là. Non, je reste connecté, mais je ferme mon micro et ma caméra. Ça va?
Parfait.
On fait comme
Merci.
ça. C'est simple, hein. faut réciter des fables de La Fontaine. Ça, c'est ce que je fais quand je suis stressé. Vous pouvez rallumer votre micro, comme ça j'entends un peu ce qui se passe, c'est plus agréable. Voilà, c'est juste pour avoir un peu de bruit de fond, c'est plus agréable.
Bien sûr, pardon.
C'est Jean-Personne?
Just to make sure that also remote people hear the questions, which is maybe unlikely. Just repeat the questions and it will be fine.
Ok.
Voilà, c'est juste pour avoir un peu de bruit de fond, c'est plus agréable.
Non, non, c'était bon. Je crois que ça a répété parce qu'il doit avoir le lien YouTube ouvert en même temps et il y a un petit décalage.
Oui, c'était ça, mais je viens de couper le son. C'est bon.
Merci.
Tu l'as connecté à la salle?
Non, c'est pas possible.
Ah oui.
I think we can start.
Alex, c'est celui qui a officié les précédents.
Wait, because I need to find the list of the members of the jury with all the affiliations.
You mean the thing that's in front of there?
It's actually much easier than I anticipated. I already have a PhD, so it's okay.
It's just a reading test.
Okay. All right. So I will act as the president of this PhD committee for the PhD defense of Maëliss Jallais. So in front of the jury that I will present now. So we have two reviewers. Marco Palombo, who's an associate professor at Cardiff University, who is here. Jean-Philippe Tirand, who is remote, he's professor at the IFR in Lausanne.
Good afternoon.
We have Jelena Jelescu, who is assistant professor in Lausanne University Hospital, also in Lausanne. Derek Jones, who is a professor in Cardiff University, the director of the Kubrick Institute. Dimitri Novikov, associate professor at NYU, and Julien Valette, who is a research director at CEA, which is just nearby. And I'm Alexandre Bonfort, I'm a director de recherche at INRIA in this building. So now, Maëliss, you have 45 minutes to present your work, which will be followed by questions. First question from the reviewers. We will start with Jean-Philippe afterwards, so far away. And then we have all the questions from all the examiners. The floor is yours.
Thank you for the introduction. I would like to thank the members of the jury for being here today. I'll be presenting my PhD thesis work entitled Enabling Cortical Specific Sensitivity on Diffusion MRI Microstructure Measurements using Likelihood-Free Inference. This work was done within the parietal team at INRIA under the supervision of Damien Wasserman. The brain is a central human organ for taking decisions. It can be decomposed into two main parts. The outer part, in purple here, is called the gray matter and contains mainly neurons. The lighter part in the center corresponds to the white matter. It's composed of long-range myelinated axons, which connect different regions of brain-gray matter together. Brain organization can be altered by neurodegenerative diseases, which lead to dramatic consequences. Symptoms include a progressive and irreversible loss of mental functions, and particularly memory. Dementia severity and cognitive deficits appear to be mainly related to neuronal loss and synaptic pathology. Being able to quantify changes at the neuronal scale seems crucial to understand those neuronal diseases and their evolution. Neurons being mostly present in brain-gray matter will focus our study on this part of the brain. To measure the tissue microstructure, such as the proportion and size of the neurons, the developed technique should be non-invasive so that it can be applied on humans in vivo. It should also provide a global view of the brain and allow for studying its evolution through time. Given these constraints, diffusion MRI appears to be a perfectly adapted method for tissue microstructure quantification. Diffusion MRI is based on the study of water molecules movements. Indeed, those molecules constantly undergo random microscopic movements known as Brownian motion. In this example, molecules are freely diffusing over the space. When no barrier impedes diffusion preferentially in one direction over another, molecular displacements are equal in all directions, which is known as isotropic diffusion. However, in the brain, molecules' movements are hindered, including by cell membranes. Diffusion is then not equal along all directions anymore and becomes anisotropic. The distance traveled by a water molecule depends on its interactions, in particular with other molecules and cell membranes. Certain geometric characteristics of the underlying structure at the microscopic level can therefore be inferred from the molecule movements. I will now present how in practice we can capture a signal that relates to these molecule movements. Consider an MRI acquisition sequence. A magnetic resonance scanner is constantly emitting a homogeneous magnetic field referred to as B0. During an acquisition, a magnetic field whose strength varies linearly along a specific direction is emitted. This magnetic field is called the gradient. Nuclei along the gradient will change their spin frequency in a predictable way. we say that they are processing. A radio frequency pulse is then generated at a particular frequency, making some spins resonate and allowing to select a slice. In order to retrieve the signal generated at each position within this slide, 2 more gradients are needed. A phase and a frequency encoding gradients are successively turned on that enable to encode the signal in the K-space. We can finally decode the MRI image by taking the Fourier transform of the K-space. To obtain a diffusion image, gradient pulses are added to the acquisition sequence after the plane selection RF pulse. In this thesis, we focus only on the pulse gradient spin echo sequence. Let's consider the selected slice after the radio frequency pulse. All the nuclei of this slice are processing at the same frequency W. In a PGSE sequence, a first gradient field G is applied, making the nuclei process at frequencies which vary linearly in the right direction. In this example, nuclei in the top row process at the frequency W1 and nuclei in the bottom row at the frequency W2. After a time big delta, we apply a second gradient with the same strength but in the opposite direction during the same amount of time. This gradient cancels the phase shift induced by the first one, and we come back to the same situation as at the beginning. However, in practice, after turning the first gradient on, molecules are randomly moving, which is the Brownian motion I mentioned earlier. If molecules are not at the same location, the second diffusion gradient changes their phase differently. Nuclei processing at different phases on the same section of the slice will cause a destructive interference, resulting in a loss of signal. The ratio between the signal obtained with diffusion gradients and the one without them quantifies the amount of ongoing diffusion. The further a molecule travels from its initial position along the gradient direction during the time big delta, the greater signal attenuation we get. The objective is to deduce the structure of the medium where the water molecules are trapped in from those molecular movements and from the loss of signal. The PGSE sequence is usually represented by this graph where we have 2 gradient pulses of strength G and applied during a duration small delta and separated by a time big delta. We express the diffusion time as big delta minus small delta over 3. The diffusion weighting is globally encoded by the Q value. It reflects the strength and timing of the gradients used to generate the diffusion weighted images, where gamma is generally the nuclear geomagnetic ratio of the water proton. Large Q values allow to study smaller molecular movements. The B value can also be used to describe the diffusion weighting of an acquisition. It's directly related to the Q value and the diffusion times. I will use both the B value and the Q value formalisms interchangeably in this presentation. So given a multi-shell diffusion MRI acquisition, we aim at estimating brain gray matter microstructure. In other words, for each voxel, we have signal values for multiple Q values acquired in multiple directions. The Q values small delta, big delta, and small delta are known. From this information, we want to notably estimate the size and the density of the neurons in the brain gray matter. The proposed method is based on an approach called tissue modeling, which relies on 2 main steps. First, we need to define a forward model based on the biophysical modeling of brain gray matter. This model helps define a relationship between the tissue parameters representing gray matter tissue microstructure and the diffusion MRI signal. Once we have defined this model, we need to solve the inverse problem of retrieving the tissue microstructure from the diffusion MRI acquisition. In the pursuit of this objective, I'll first cover the biophysical model used in this thesis, which is an impermeable 3-compartment model with SOMA sensitivity. I will then present the inversion method that we developed, which is based on likelihood for inference. They correspond to the 2 main contributions of my PhD thesis. I will then go over some limitations and future directions. First, I'll introduce the forward model used to describe brain gray matter and show how we extract some summary statistics of the diffusion signal from it. So this forward model is an impermeable 3-compartment model with SOMA sensitivity. This work was first introduced at ISMRM in 2020. In order to define a biophysically valid model, we need to study the composition of brain gray matter. Research in histology has shown that brain brain gray matter is mainly composed of neurons embedded in a fluid environment called the extracellular space. Each neuron can be subdivided into a soma, which is the cellular body surrounded by branches called neurites. Brain matter has been widely studied during past decades. A legitimate question is, can we use the same model for both the gray matter and the white matter? To answer this question, let's have a look at the popular white matter model, which is a two-compartment model called the standard model. Brain white matter is mostly composed of axons. These axons can be modeled as a collection of cylinders. Their radii are considered negligible compared to the diffusion lengths a molecule can travel in the longitudinal direction of the cylinders for a given diffusion time. These cylinders with zero radius are commonly referred to as sticks. Molecules are only diffusing in the parallel directions of the stick with a diffusivity dA. The extra-axonal space is modeled as a sum of local anisotropic Gaussian contributions with parallel and perpendicular diffusivities dE. Exchanges between intra and extra-neurite water are generally neglected, notably due to the myelin sheath surrounding the axons, making them impermeable at the timescales used in clinical diffusion MRI. The resulting diffusion signal in brain white matter is then modeled as a sum of anisotropic compartments, which can be written as a convolution between a response kernel K from a perfectly aligned fascicle pointing in the direction N and the fiber orientation dispersion function P. Recent evidence shows that the standard model does not hold for gray matter. Several assumptions have been proposed to explain this issue, such as the abundance of cell bodies in gray matter, increased permeability in neurite membranes, or structural disorder leading to non-Gaussian diffusion along the neurites. In this thesis, we follow the hypothesis that the standard model does not hold due to an abundance of cell bodies in gray matter. We therefore modeled brain gray matter using a three-compartment model similar to the model SANDI. We follow the assumption that our protocol is insensitive to exchanges between the compartments, that is, that molecules moving from one compartment to another have a negligible influence on the signal. The three compartments are the neurites, which are modeled as sticks, similarly to the axons in the white brain matter, somas, which are modeled as spheres, and the extracellular space, which is modeled as isotropic Gaussian diffusion. The observed diffusion signal is considered as a convex mixture of signals arising from those three compartments. Fn, Fs, and FECS are the signal fractions of neurites, somas, and the extracellular space. These relative signal fractions do not correspond to the relative volume fractions of the tissue compartments, as they are also modulated by different T2 values. The other parameters of interest in this model are diffusivities within each of the three compartments and the soma radius. All these parameters are orientation independent, which enables us to work on the direction average dim MRI signal, denoted S bar, also known as the powder average signal. This consideration mainly matters for neurites, as their signal is not isotropic, unlike the proposed models for somas and ECS. We now review the model for each compartment to make explicit the impact of each parameter on the diffusion MRI signal. As in the standard model, neurites are modeled as sticks with effective diffusion along the parallel axis and a negligible radial intraneurite diffusivity. Its diffusion signal can be written as a convolution between the response kernel K from a perfectly aligned fascicle pointing in the direction N and the fiber orientation dispersion function P. Its direction average signal follows a parallel. We aim at estimating two tissue parameters linked to the neurites: Fn, the proportion of signal coming from the neurites, and Dn, the parallel diffusivity. Somas are modeled as spheres and follow the Gaussian phase distribution approximation. We exploit this relation to extract the parameter Cs, which at fixed diffusivity is modulated by the radius of the soma Rs. Using this parameter instead of Rs and Ts separately allows to avoid indeterminacies, as I will show later in the presentation. However, a drawback of this parameter is that it's usually harder to interpret. The extracellular space is approximated as isotropic Gaussian diffusion, that is a mono-exponential diffusion signal with a scalar diffusion constant dE. This approximation assumes that the ECS is fully connected. We considered dE nearly constant per subject acquisition and estimated it as 1/3 of the mean diffusivity in the subject's ventricles. We model brain gray matter with 3 compartments, each represented by simple geometric shapes whose diffusion signals have analytical equations. This allows to have a relationship between brain gray matter tissue microstructure and the diffusion MRI signal. We then need to solve the inverse problem of retrieving those tissue parameters from a DMRI acquisition. The acquired signal has a large dimensionality. Solving the inverse problem directly from the direction average signal equation leads to indeterminacies, that is the existence of multiple mathematical solutions. We then end up with several tissue parameter values that could explain equally well the observed diffusion signal. It can be very complicated to disentangle the correct parameter combination that actually generated the acquired diffusion signal. To tackle this problem, some methods suggest to stabilize the solution by imposing constraints on model parameters, such as the model NODI. However, the validity of those constraints need to be questioned. They suggest, for example, that diffusivity within the neurites and along the parallel direction of the ECS are equal, and that they are the same for all voxels within brain white matter. Diffusion-weighted spectroscopy seems to indicate the contrary. Also, if we admit that the equalities are correct, they imply that a small deviation from the fixed values, as occurs in cerebral ischemia, will induce a non-negligible bias in the other parameter estimation, leading to false interpretations. Moreover, in spite of those constraints, the inverse problem remains largely degenerate. Some constructing methods take a different approach by relying on very demanding acquisitions with many Q shells, including very large p-values. These requirements are not feasible for clinical applications on humans because they involve very powerful gradients and for the patients to stay a very long time in the diffusion MR scanner. At last, some methods propose to reduce the dimensionality of the data to be processed, such as the lemonade system of equations. This solution seemed the most promising, and we extended this idea to our 3-compartment model. We decided to follow the idea of reducing the dimensionality of the data by extracting summary statistics from the diffusion MRI signal. These summary statistics are rotationally invariant features directly related to the tissue microstructure parameters. They are obtained from a signal analysis based on the large and the small Q-value approximations that I will present now. The small Q-value analysis that we named Spike Lemonade is extended from the lemonade method developed for white matter. It's based on the cumulant decomposition, which is then factorized in the spherical harmonic spaces. This allows us to extract 4 rotational invariant scalars, and we can note the appearance of a parameter P2, which measures neurite orientation dispersion. For perfectly aligned neurites, P2 equals 1. It equals 0 for a fully isotropic distribution. The original method suggests to use p-values compressed between 0 and 2.5 milliseconds per micrometer square because the cumulant series is expected to converge in this range. Moving to the large Q-value analysis, we compute a Q-bounded return to the origin probability, which is a measure of the restrictions of the diffusing fluid molecule motion. From the Q-bounded RTOP equation obtained from our 3-compartment model, we can extract 2 additional scalars, A fit and B fit. You can see what's happening more practically in the figure. For Q large enough, the Q-bounded RTOP coming from the soma is converging towards a value that depends on the radius of the somas, while the neurite RTOP is diverging. You can also notice the addition of a gamma times Q cube term, which was added experimentally to capture excess noise. We thought it was only capturing this noise in the direction average signal, but we discovered it could also be capturing edge changes between the neurites and the ECS. As a summary, we are willing to estimate a parameter theta, which contains 6 tissue parameters of interest. We have the parallel diffusivity of the neurites dn, a proxy to soma size called CS, the neurite orientation dispersion P2, and the signal proportions of our 3 compartments. After the small and large Q-value analysis, we also end up with a unitless vector X, which contains the normalized summary features, with DE estimated beforehand as 1/3 of the mean diffusivity in the ventricles. They are related by a transformation M, such that X equals M of theta. The goal is now to estimate a theta 0 given an observation X0. The resolution of this inverse problem corresponds to my second contribution. We propose a solution to solve this inverse problem based on likelihood-free inference. This work was presented as oral presentations at ISMRM and IPME in 2021. It has also been submitted to the journal Melba. Most of the methods are solving the inverse problem by casting it as the least square problem. Palumbo et al. Suggested to employ random forest regressors. Although an acceptable accuracy is obtained in real cases, these techniques, as used in the literature, only deliver one possible set of tissue parameters, masking therefore other biologically plausible solutions that could explain the observed signal. No description in terms of confidence into the output parameter estimates is returned. The challenges are then to be able to obtain not only one value, but a full posterior distribution over the parameter space. This could point out degeneracies and allow to define a confidence factor. To that aim, we propose a probabilistic framework to solve the nonlinear inverse problem efficiently using Bayesian inference. The general framework is the following. For each voxel, we have a diffusion signal acquired for multiple Q values, each Q value acquisition being done in many directions. We extract summary statistics from this multi-shell signal for each voxel separately. We then solve the inverse problem using a Bayesian method called likelihood-free inference or simulation-based inference, which produces an approximation of the full posterior distribution. This can be used to determine which microstructure parameters are the most likely to have produced the acquired diffusion signal, the confidence that we have in this estimation, and also detect indeterminacies. The question now is, how can we obtain the posterior distributions denoted P of theta given X? For a given observation x0, Bayes' theorem gives us the following formula, where P of x0 given theta is the likelihood of the observed data x0 given theta. P of theta is a prior distribution describing our initial knowledge of the parameter values. In practice, it usually corresponds to the physically possible intervals of the parameters. And P of x0 is a normalizing constant commonly referred to as the evidence of the data. This equation allows to easily and efficiently compute the posterior distribution of theta given an observation. However, the likelihood function of complex models, such as the one that we consider here, is often very hard to obtain and makes the Bayesian approach rather challenging to use. Likelihood-free inference bypasses this bottleneck by approximating the posterior distribution using a conditional density estimator parameterized by phi. In practice, this is done in two steps. First, we generate a set of n paired samples, theta I xi, where theta I follows the prior distribution and xi is obtained from the forward model presented before. Our posterior approximation is then obtained by minimizing its average Kullback-Leibler divergence with respect to the conditional density estimator for different choices of x. This means that we're looking for the conditional density estimator Q, which is the closest to the true posterior distribution P in the KL sense. This conditional density estimator is parameterized by a parameter phi. After developing the loss function, we use in practice the Monte Carlo approximation to the Kullback-Leibler divergence. It's possible to show that the minimizer of the loss L converges to P of theta given X when n tends towards infinity. In this work, our conditional posterior distribution function approximators belong to a class of neural networks called normalizing flows. Normalizing flows are invertible functions capable of transforming vectors generated by a simple Bayes distribution, such as the standard multivariate Gaussian distribution, into an approximation of the true posterior distribution. These flows allow to construct flexible probability distributions by pushing an initial density through a series of transformations to produce a richer, more multimodal distribution. These transformations are universal approximators, which means they can approximate any posterior distributions without needing to know in advance the shape of the posterior distribution you're trying to estimate. We use an autoregressive architecture for normalizing flows implemented via the masked autoencoder for distribution estimation. We follow the same setup from Greenberg et al. For likelihood-free inference problems, stacking 5 mates and using a standard normal Bayes distribution. This is a standard choice of implementation in the literature. It provides a sufficiently flexible function capable of approximating complex posterior distributions. To summarize, the general pipeline that we use for each subject is the following. We start by estimating DE as 1/3 of the mean diffusivity in the ventricles. We then run simulations of paired samples theta I, xi, following the 3-compartment model presented before. For each voxel of the brain gray matter, we then extract summary statistics from the diffusion MRI signal. And at last, we compute the posterior distribution by minimizing the loss function. Before applying the method to real datasets, we performed a series of tests to assess the pipeline worked correctly on a setting where the ground truth parameters were known. We used the software package DMIPy, which is a simulator for diffusion microstructure imaging in Python. We use it to generate each compartment individually and then sum them with proportions Fs, Fn, and FVCS. We also use the SB8 package for carrying the likelihood for inference procedures. The prior distributions are defined as uniform distributions with limits within physiologically relevant intervals for each parameter. Signal proportions are all comprised between 0 and 1. We follow the usual assumption that the diffusivity of the compartments are inferior or equal to the self-diffusion coefficient of free water, which is 3 micrometers squared per millisecond. The range for CS has been computed for radii between 2 and 30 micrometer and the same diffusivity range than for TN. At last, P2 is comprised between 0 and 1, with 1 indicating an isotropic orientation dispersion. We used 100,000 simulated data points to approximate the posterior distribution. While we have validated the likelihood for inference pipeline on multiple choices of physiologically relevant ground truth parameters, we present the results for only one of them. Note that in the simulations, with all setups, we have used the 3 B-shells with the lowest B values for the spike lemonade approximation and the 3 largest Q values for the RTOP approximation. First, we wanted to confirm whether the use of summary statistics for the GMRI signal actually conveyed any improvements in the parameter estimation. We consider the setup that we dubbed ideal because it corresponds to a comfortable case with 10 B values between 0 and 10 milliseconds per micrometer squared. We use the same acquisition times as in the HTTP MGH database. The ground truth values that generated the observed signal are marked as vertical black dashed lines. We have considered 3 different cases. In case 1, we generated X0 directly from θ0 using the forward model. This is a rather favorable case for a posterior approximation since it's applied on a data point generated in the same way as the dataset in which it was trained. We see that in case 1, the marginals of the posterior distributions are peaks well concentrated around the values of the ground truth parameter θ0. This confirms that the posterior approximator successfully inverts the nonlinear model using the examples in the training set. In case 2, we generate the GMRI signal S0 from θ0 using DMIPy and then extract the summary statistics from the GMRI signal to obtain X0. We use the same posterior approximator from case 1, meaning that the data point in inference time is generated differently from those in the training procedure. This is the actual realistic case that interests us the most. The parameter estimation in case 2 captures very well most of the true values of the parameters as we obtain sharp distributions with a small bias in parameter estimation. It indicates both that our posterior approximator is robust to observe data generated differently from its training sets, and that our summary statistics are descriptive enough to synthesize each tissue compartment. In case 3, we generate a GMRI signal S0 from θ0 using DMIPy and do not use summary statistics for the model inversion. We directly feed the signal, that is in this case, we consider X0 equals S0. The posterior approximator was trained this time on a dataset with observations consisting of GMRI signals, so it's different from the approximators in case 1 and case 2. The posterior distributions for this case are wider, indicating that more combinations of tissue parameters could have explained the obtained diffusion signal and that we can less trust the results. These posterior distributions are almost flat for P2 and DN, indicating that the likelihood for inference method is not capable to estimate the ground truth values for these 2 parameters. These poor results reflect the largely indeterminate system of equations that prevents us from trying to infer the tissue parameters directly from the GMRI signals. This behavior was expected as similar degeneracies was shown for a simpler model with only 2 compartments. We also questioned the utility of the parameter CS that we introduced as a proxy of the SOMA radius. We wondered whether there was a real benefit of using the parameter CS instead of estimating the radius RS and the diffusivity DS directly. This figure shows that several radius diffusivity pairs correspond to the same CS value, with an altered or likelihood for inference pipeline, so to consider an extended parameter vector, including RS and DS. We obtain large marginal posterior distributions for each parameter. The joint posterior presents a valley in green of RS-DS values that could explain the observed summary statistics with high probability. The set of solution includes the ground truth value, which is marked with a red dot. Estimating SOMA radius and diffusivity separately leads to indeterminacy. Estimating CS instead allows to avoid this problem. We have also investigated how different choices of p-values in the acquisition scheme affect the quality of the parameter estimation. Note that these choices have no influence on how the posterior approximator is obtained, since it's trained on data generated directly from the relations between tissue parameters and the diffusion summary statistics, in which the p-values do not interact. The p-value choices only have an impact on the obtention of the summary statistics. So summary statistics are obtained from a small q-value approximation, where p-values are expected to be between 0 and 2.5 milliseconds per micrometer square in order for the moment decomposition to be valid. The large q-value approximation is based on a q-bounded RTOP. This figure presents the RTOP signal obtained from spheres with different radius. We can observe that the bigger the spheres, the sooner in terms of q-value, the signal would converge towards a plateau, which is related to the summary statistics A-fit. So the large q-value approximation for big SOMAs requires lower q-values than for small SOMAs. Consider, for example, a SOMA radius equals to 12 micrometer. If q is inferior to 70/1/meter, RTOP of the SOMA will not have converged, leading to a bias in the parameter estimations. We carried out our simulations on DMIPy considering 3 acquisition setups. For each setup, we generated a signal, extracted summary statistics, and computed its posterior distribution. All setups have p-shells with 128 uniformly distributed directions, but they differ in their p-values and acquisition time. In blue, the setup ideal corresponds to the comfortable case we used in the previous simulations, with 10 p-values. We use the same acquisition times as in the ATP-MGH database. As previously, we see that estimations in the ideal setup are very much concentrated around the true values of the parameters. Setup ATP-MGH reproduces the setup from the ATP-MGH database with 5 p-values. In this setup, the estimations are rather good, allowing us to apply the method onto real data and to expect to have stable results. Setup ATP-1200 reproduces the setup from the ATP-1200 dataset with only 4 small p-values. Note that the acquisition times are different compared to the 2 other setups, leading to another ground truth for the parameter CS, which is indicated as a vertical dashed green line. A bigger bias is present for this setup, as expected from the extraction of the summary statistics in the large q-value approximation. We therefore expect the estimations on real data to be more biased and more unstable in the ATP-1200 dataset than in the ATP-MGH dataset. After validating our proposal on different simulated settings, we carried out our analysis on the ATP-MGH dataset, which is composed of 31 subjects. For each subject, we estimated the ECS diffusivity in the ventricles. We then ran the inference and segmented the brain gray matter using free surfer. For each segmented voxel, we then extracted the summary statistics, computed the posterior distributions of the tissue parameters, and then extracted the mean estimation along with a confidence value. We deemed an estimation stable with the mean values are larger than 2 times the standard deviations obtained via the likelihood for inference pipeline. Per subject, the inference takes approximately 1 hour using 20 CPUs, and the estimation of the seven parameters for every voxel in the gray matter is about 3 hours. Each subject was then registered to the MNI space. We averaged the results and projected them onto an inflated surface. We masked our results to show only areas where parameters were deemed stable, indicating that our posterior distribution is narrow and centered around its mean value. We observe a lack of stability on small sections, including the auditory cortex and the precentral gyrus fundus. Our figure assesses qualitatively the results on soma size by comparing with Nisselstein histological studies. Our comparison shows good agreement between different cortical areas and the parameter CS. Interestingly, most regions in which the parameter estimation has low confidence are located in the fundus of the sulci. Two main hypotheses could explain such behavior. Firstly, brain regions which are very curved may be more prone to mixing between tissue layers and the cerebrospinal fluid, which generates noisier signals. Thus, the estimation of summary statistics become more biased and the posterior distributions tend to be wider. Another possible explanation, based on cytoarchitecture considerations, points out to the fact that the fundus of sulci is where sharp changes in the cellular populations happen. A mixing of several types of neurons in one voxel could generate multimodal posterior distributions and hence a region with large variance. We computed the mean soma signal proportion in the cortex using only trusted estimations. This mean equals 0.22, which is coherent with the mean volume occupancy of 10 to 20% observed in gray matter. To interpret the results at a region-based level, we have extracted mean values from 12 regions of the Brodmann Atlas. A clear difference in soma proportion can be observed in those regions, which seems coherent with histology. These results are then very encouraging, although more experiments would be required to confirm for general agreements between our estimations and the microstructure composition of brain gray matter tissue. Despite the CS parameter being useful for evading indeterminacies, its biological interpretation remains difficult. With the goal of relating our CS estimation with physiological insight, we estimated the soma ranges by fixing soma diffusivity. Similarly to the model Sandy, we fixed soma diffusivity to the value of self-diffusion coefficient of free water. This value could be adjusted for each voxel and is only used here in a matter of comparison and interpretation. Using a fixed point method, we computed the soma ranges from the average CS mapped obtained from our posterior distribution. We see that the estimated soma ranges vary between 8 and 14 micrometer, which is in accordance with histology. We applied the exact same pipeline on 30 subjects of the HCP 1200 datasets. We see that 55% of the CS estimations on brain gray matter is considered unstable and is therefore masked. Indeed, our Q-banded RTOP approximation relies on a high Q value, where the signal coming from the somas is expected to have converged. The larger the soma, the sooner the Q-banded RTOP will converge. However, the largest B-value contained in this database equals 3 milliseconds per micrometer square, which is not enough for the signal to have converged. The low B-values combined with the noise present in the acquisition leads to a poor quality of the extracted summary statistics, which itself leads to rather wide posterior distributions, resulting in unreliable results. Note, however, that estimations of the superior temporal gyrus are not masked, and both datasets seem to indicate large neurons in that region. The proposed approach has been designed and applied to brain gray matter, but one could also want to apply it to other brain regions. We decided to apply it to brain white matter. Results on the HCP MCH datasets indicates that the axons distribution is more anisotropic in white matter than gray matter, with values coherent to the ones obtained with the Lemonade framework. Somas are also less present in white matter than gray matter, as indicated by a lower signal fraction Fs, which is expected from histology. However, the ECS model used in the three-compartment model defines its diffusivity as isotropic, while the tensor representation is usually preferred in brain white matter. The application of a similar likelihood-free inference pipeline based on a model designed for white matter could help improve the estimations of tissue parameters and better interpret its output thanks to the posterior distributions. For example, this pipeline could be a new way to solve the lemonade system of equations within a probabilistic framework. The three-compartment model that we use approximates well the intracellular signal in brain gray matter by adding a sphere compartment to account for soma presence. Other explanations have been proposed besides the abundance of cell bodies in the gray matter. We expanded the three-compartment model by also accounting for an increased permeability in neurite membranes. That is, we also modeled the exchanges between neurites and the ECS. This extension corresponds to my third contribution as a co-supervisor, which was recently accepted to ASMR. The tissue microstructure parameters are the same, except that we added an exchange time Tx. The resulting diffusion MRI signal is considered as a weighted sum between the signal coming from the exchanging compartments, modeled using the Kerger model, and the signal coming from the non-exchanging ones, that is the somas. We extracted summary statistics from a large and small Q-value analysis and obtained some preliminary results on simulations. To summarize, we proposed an impermeable three-compartment model with soma sensitivity, extracted summary statistics using a large and small Q-value analysis, and solved the inverse problem using a likelihood-free inference-based algorithm. We also extended the model to also account for exchanges between the neurites and the ECS. The main advantage of our method is its capacity to estimate a full posterior distribution. This one allows us to assess the quality of the posterior of the parameter estimations, to characterize regions where it's harder to invert the model, and to avoid indeterminacy. We also alleviate limitations from current methods by not fixing parameters to predefined values. The code is available in GitHub, which allows to simulate and solve the inverse problem for both models. I would also like to mention the last publication, which is a book chapter which was published in 2021. This concludes my presentation. I'd like to thank my advisor, Damien Wasserman, as well as all my collaborators and colleagues from Python. Thank you for your attention.
Thank you very much, Maëliss, for this presentation. We now have the questions from the jury. And so we will start last time with Jean-Philippe Tirand, who's remote.
Yes.
People will be able to hear Jean-Philippe. If it's not the case, just feel free also to repeat the questions.
So you can hear me, right?
Yes, we can.
Yes.
Very good. So thank you very much, Mr. President, first. And most importantly, thank you, Maëliss, for the invitation, and thank you, Damien, for the invitation to be part of this jury. I really appreciate. And although I cannot see you, I would like also to say hello to the other jury members. I really apologize for not being here with you or there with you today in Paris. So I hope I will see you in the deliberation. So first, let me congratulate you, Maëliss, on this excellent oral presentation first. Also on the manuscript, which was very complete, contains actually more than what you presented, or at least more details than what you presented today. And it's extremely clear, complete, very rigorous, very, dense in some sense, but also remains very readable. And in general, it's a very good contribution in terms of, things that people will want to read in the future. And also, of course, congratulations for the work itself, which is really outstanding, especially the focus that you have on estimating the parameters, also with the confidence intervals and the identification of indeterminacies, which is really of particular interest. So congratulations for that.
Thank you.
That was my introduction. So now questions. So I have a few questions on your two main chapters, 4 and 5. And I will try not to be too long to leave enough time for the other jury members. You have a lot of jury members and very much expert in the field, so I will try not to be too long. Okay, the first questions on chapter 4, on your two models, basically. First, I was curious to see that you don't really discuss the permeability of the soma itself. You discuss it, but a little bit marginally. And I was wondering, what do we know about that? Is it a significant part of the exchanges, or is it negligible due to its restricted surface? And what is your feeling on how important it could be with respect to the permeability of the nerites themselves?
First, thank you for all the nice remarks. Then concerning the somas, the general hypothesis which is made is that indeed its surface is very small compared to the surface of the nerites, and that the exchanges can therefore be negligible compared to the nerites.
Yeah, but at some point in your manuscript, you say that it's not really the case, right?
Yes, so I'm gonna show an extra slide.
We didn't prepare that, right?
So it could be non-negligible anymore in the case where we have a very high exchange time. So indeed, for example, in one example that we took, the volume to surface ratio was 10 times bigger for the nerites than the somas. This ratio can be directly related to the exchange time, Tx. And so for a fixed extrasolar space, then the Tx of somas equals 10 times approximately the Tx of nerites. So this means that if Tx of nerites is quite large already, then the Tx of somas can be considered too big to really have an impact. And so we consider the somas as impermeable. However, if the Tx of nerites is really slow, then the Tx of somas could be taken into account.
Okay, thank you. That's useful insight indeed. Maybe a second question on something that also is present in your description of the models, but a little bit marginally, is the extracellular space compartment, and especially about the value of the diffusion coefficient dE that you have for this extracellular space. In our simulation, we kind of see a very strong influence of the value of that parameter on the signal itself, especially at short diffusion times. Did you experience the same? And how can you comment on the importance of having either measured or precise estimates of that parameter?
So concerning that parameter, so we are estimating it beforehand, so in the ventricles, so that we can get rid of it in the parameter estimations while solving the inverse problem.
That I agree. My question is really whether this value is the good one, and if it's not, it might have a strong influence on the signal and hide much of your microstructure.
Yes, that's one of the main limitations, I think, of our model, is that the Gaussian approximation that we make might not be correct for the gray matter. So several extensions could be thought of. For example, we could model it similarly to the standard model, where we have a parallel and a perpendicular dE values. Otherwise, I don't exactly know how. I guess more experiments would be needed to define exactly which model would work better for the ECS. But yes, that's definitely one.
And it would remain tractable if you introduce this as an unknown. Would it remain tractable, or would it introduce more indeterminacies?
We would require to estimate more summary statistics from the signal approximations, so that the whole system can still be inversible.
Okay, good. Maybe a question about chapter 5 now, and about your famous CS coefficient. So that you introduced to cope with indeterminacies. So obviously, you clearly show that it helps and it allows to avoid those indeterminacies when you invert the model. But at some point, if I want to be a little bit, a little bit picky, those indeterminacies, they come back when you estimate the actual values of the tissues, because since your coefficient is a function of both the size of the cells and the diffusion, diffusivity, at some point you have to mitigate that and do like Marco in his work, fix the value to try to get that. So at the end of the day, I'm wondering what is the specific advantage of introducing that parameter if at the end of the day you still need to fix one of its contributions to estimate the other, and how does it compare to the work of Marco Balombo himself, where he managed to have very good estimates of the sphere diameters without having this parameter. So how do you compare those two works and your specific advantages?
Yes, so by fixing the diffusivity in the somas, then you're only acquiring one RS. Our parameter CS allows you to try different diffusivities according to extra knowledge that you could have, and therefore gets multiple possible values of radius depending on your model and your assumptions. Also, another advantage of the CS parameter, let me show that, is that you have two different regimes to, I would say, two simpler interpretations in two border cases. You could relate CS directly to DS, and also in the wide post regime, you could approximate CS directly to RS. So in our databases, we were not in those cases. However, if you are, for example, in the Newman regime, then the CS directly is proportional to the RS, and so you would get that value directly. Okay.
Yeah, I think I'm pretty sure other members will come back to this discussion. So let me continue. I have a question about the simulations that you have done. I think it makes perfect sense. Still, they're done in rather simple conditions, in the sense that you simulate from the equations that you have used to derive the model. And I was wondering whether it would make sense to simulate on more realistic or more complex substrates using other types of simulations, maybe, just to try to see how robust your estimation is in more realistic conditions, more geometrically realistic conditions. I don't know if you see my point. Can you elaborate on that?
Yes, exactly. So it's an ongoing work that we are actually doing. We're using a simulator based on the finite element methods, and then we're trying to compare the obtained posterior distributions with the ones that we have. However, so far we have a few limitations. I have another extra slide. So here, for example, we simulated a neuron directly from neuromorpho and generated the signal. And we observe differences with our models. We have possible explanations for why it's not working. So one of the main hypotheses is the problem from the ECS that you were mentioning before. Okay. In those simulations, it's not a Gaussian diffusion. Yes. And so more work would be needed, both to make the simulations more accurate, and secondly, improvements in the model itself.
And also maybe varying the density of the cells, the number of spheres that you have. It might be the size of the substrate, which also has an influence on the quality of your simulation. So that's very exciting perspectives indeed. Okay, maybe one or two more questions and then I will be done. Okay, what you presented here at the end, which is the first steps towards the estimation of the parameters for the permeable membrane model. So you showed very interesting preliminary results on the indeterminacies and on the robustness of the estimate. What I just would like to see is whether you also investigated the quality of the estimation comparing permeable and impermeable models, whether how much it brings, how can I say, which one brings back more robust estimates of the actual microstructure? You see what I mean?
Yes, we investigated that.
I don't think this is in your manuscript, is it?
I think,
or maybe I missed it.
Yes, I think this figure was presented in the manuscript.
Sorry. Yeah, maybe I missed it.
It shows the impact of the permeability in Neuric membranes compared to the model without the permeability. And we see that it can have an influence up to 30% in realistic cases of TX superior to 20 milliseconds.
But what is the impact on the accuracy of the estimation itself of the microstructure parameters?
We did not test that yet, but it's ongoing work.
Because that's the ultimate question, you know. At the end, you want to estimate the parameters. Yes. So having a more realistic model, which includes permeability, should ultimately lead to a better estimate of the parameters. But the cost is that the model becomes more complicated and potentially more with more indeterminacies and so on. So that's the key question. How far do you want to go in realistic models until they become intractable?
Yeah, agree.
Okay. Ali, one more. Okay. Yes, maybe one or two last ones, more general. So you also investigate your methods for other types of tissues, and you consider the cerebellum. Naive question: is it really another type of tissue? Isn't it very close to what you actually have? That's the first part of my question.
Yes, actually, the cerebellum, you have a white matter part and a gray matter part.
Yeah, exactly.
So yeah, it's
okay. So what about other types of tissues in the brain? So I don't know, deep brain structures like amygdala, which have a structure that could be of interest. I don't know, in some dementia or something like that, you might want to estimate the cell densities in that. Did you consider that at some point? And even looking further, what about non-cerebral tissues, other types of tissues? Whether your model would still kind of course, you will not have neurites, but still the SOMA model or kind of could be very attractive. So can you elaborate on that?
Yes, that is a really good question. We haven't tried that. However, it would be a good way to go to continue to analyze the posterior distributions that we have in different tissues and being able, also the main goal would be to have ground truth values to be comparable with. Sure, so maybe in those different regions, it could be easier than the gray matter that we're doing.
Probably, yeah. Biopsies are probably more feasible and things like that. Very last question. So when you estimate your parameters, you have this kind of confidence interval. Can you relate that to the variations that you expect for those parameters in case of pathologies? Because your introduction, you clearly mentioned that those techniques will be useful to study, I don't know, brain degeneration, neurodegenerative disease, whatever, which by hypothesis have a loss of some kind of cells, of neural cells. So can you relate, in one example that you might have in mind, I don't know what, the uncertainty on some of the parameters with the difference that you would expect to detect a pathology? Do you see what I mean?
Yes. So I guess we would first need to find out another confidence value, because so far we're assuming the posterior distribution is Gaussian as we're using only the mean and the standard deviation. It's possible that in non-healthy tissue, we find non-Gaussian posterior distributions, and hence we would require another confidence interval.
Especially if you have invasion by other types of cells.
For example, yes.
Imagine you have cancerous cell coming in somewhere. They might have much bigger sizes, and the Gaussian distribution might not be valid at all.
Yes, I agree.
Okay. And in general, for neurodegenerative disease, do you think that the changes in, say, in cell density is compatible with the estimates that you have, the confidence on the estimates that you have, or there is still much work to do on that?
And that's a good question. I guess
You're allowed to say that you don't know.
yeah, sure. But I guess we would probably need to slightly change the model first. And then I guess we will just need to try it and see the results. It's something we haven't done at all, but it's an ultimate goal that we would like to do.
Very exciting. Anyway, okay, I'll stop here. Thank you again, and congratulations. And thank you for those answers. That's it for me, Mr. President. Thank you very much.
Thank you very much. So now we'll have the other reviewer of the PhD to ask this question. So it's Marco Palombi, right? Yes.
Thank you very much, Mr. President. And I can only echo Jean-Philippe's gratitude to you and Daniel for inviting me here today. My greatest honor and pleasure to be here and discuss this exciting work. I found your presentation just beautiful. It was perfect from the timing, the speech, the slides themselves. You managed to cover basically everything you have done in this PhD thesis, which is a lot. And the vast majority of it is pioneering new avenues for our research field. So it's really, really exciting stuff.
Thanks.
I have some questions as well. So my first question is, during the manuscript and your presentation, you always talked about neuronal soma, neurons. But gray matter is not only neurons. Actually, neurons, volume-wise, may occupy 60% of the volume of the cellular volume. You have a lot of other cell types: oligodendrocytes, astrocytes, microglia, epithelial cells. So why do you refer only to neurons? Do you mean your technique and diffusion MRI can be so specific to select the cell population, or there are other reasons?
No, I guess so. This model is the simplest. However, like the different kind of cells, I guess they also can be modeled by the somas. I mean, we use the term somas, but in fact, it's much more than just the normal somas that you said. It's other kind of cells. So some of these extra cells have a similar model as the somas, the spheres that we model here, and others, I guess, are just considered as extracellular space. So it's a simplified model also because we cannot estimate a great number of different parameters. We need to have a simplified model, obviously, so that we have only the parameters that have an actual impact on the tissue, and that we can then estimate using the inverse problem.
So if I understood correctly, so your CS and FS do not refer exclusively to neuronal contribution. There could be some contribution from other sources, such as glial
yes.
cell bodies.
Yes.
Okay.
Yes.
Because we cannot disentangle the two.
Exactly. Yes.
Something very interesting I found when you showed the studies showing the convergence of your POP as a function of Q.
Yes.
That shows, yes.
I can put it back.
Slide number 20. This one. So you interpreted it into a negative connotation, like if it doesn't converge, then we have a bias
yes.
with our modeling.
Mm-hmm.
I would spin it around, and perhaps can we find a regime in Q and T where we can accurately and enhance the contribution of big soma, and a regime where we can enhance the contribution of your coming from small soma? Do you think this could be possible, even reformulating some of your very interesting?
Yes, that would actually be one of the goals, to define an acquisition setting where we would have the optimal Q values or B values, so that in this case it would have converged, and also would have a very good low Q value estimation, so that it would be a setup where all the conditions are united to make a perfect estimation of the summary statistics.
I agree. What I'm suggesting is, do you think a measure of deviation from this convergence could be used as a measure to introduce the fraction of small soma into your model? And so perhaps disentangle neuron and glia, which are typically very different in the cell soma size.
So if I understand correctly, you would suggest to add more summary statistics that could capture the impact of different cellular cells in the tissue. We have done more experiments concerning the CS parameter, for example. When you have different sizes of cells in the same mixing, then the CS that you obtain is the convex mixture of the two, and you don't get a multimodal posterior estimation where you would be able to disentangle different somas in the mix. No, you don't. It's only a weighted sum of those two contributions. Like you have the mean CS of the tissue. So I guess what you're suggesting would be complicated considering the summary statistics that we have here. If you model differently the different kind of cells, then maybe it would be possible, but for now it's not.
Okay, thank you. And coming to your model and Jean-Philippe observation that you effectively fix the extracellular diffusivity to a measured value subject per subject, but you do fix it. I am also concerned about the possible bias, especially in pathologies such as Alzheimer, where you have accumulation of proteins in the extracellular space that could drop dramatically. So, why did you choose DE? Not, for instance, the longitudinal diffusivity, for which you do have anyway poorer sensitivity with the conventional acquisition. That would have been anyway a value always fairly constant estimated by your model.
That is a good question. We first came up with this model in healthy tissues so that the DE could be approximated as 1/3 of the diffusivity in the ventricles. Indeed, it's not valid for non-healthy tissue. I guess we would have to change the way we estimate this DE. We also did not want to fix the DN, the diffusivity within the neurites, because we wanted to have something comparable to other kind of models, such as the standard model in white matter, and being able to compare those results in the white matter and the gray matter. Yeah, it's something we need to do, definitely. Yeah.
I mean, it's always tricky when you have to leave something out of your model, what to leave out.
Yeah.
So I'm very excited by the simulation-based inference and what really this can unlock great opportunities. However, and you actually measure the full posterior, but then you use a very, very coarse statistics to then represent the extra information you can get. And I would like just to point out that you mentioned that the random forest regressor output just one value, cannot output an interval of confidence.
Yes. So it's, yes. What I wanted to present is that in the model, for example, Sandy, if I'm not mistaken, you're only providing one result. You could get intervals, confidence intervals, but so far you do not use them in the model.
Yeah, exactly. So my question was, I came from a random forest, specifically from a random forest, I can get an interquartile range for each parameter. So how this would correlate with the variance you estimate?
The advantage of our method is that at the end you have a posterior which is not always a Gaussian distribution, and so the the standard deviation, like so far, the confidence value, we make the hypothesis that it's a Gaussian. That's one of the limitations of the method right now. However, we could define another confidence value that would explain better our posterior approximator, and not only in terms of Gaussian as the random forest regressor does, but it would allow us to see, for example, a skew or something different, or a multimodal distribution, which is not allowed in the standard random forest regressor.
I have just another couple of questions, if I can. So always on the posterior distribution, so there are GPU accelerated MCMC algorithms that allows you for these specific models, it's analytical models, so we can use MCMC methods to calculate the posterior distribution. But you didn't compare the posterior you estimate with the network-based architecture with these more classical methods.
Yes.
Is there a reason? I mean, it has been done in the past, and so we don't need to show that the method can accurately, infer the posterior, or because there is the MDT toolbox from Maastricht or Oxford that 2D mock that allows you to easily build your model and run an MCMC and get a posterior for your parameter.
Yes. I'm not really familiar with this kind of techniques, so I don't know how, in terms of time, how much time it requires to run. Also, there's the amortization question. I don't know whether you need to run a lot of simulations each time for Equation in the MCMC. In this case, you don't. You just need to run the inference once, and then you can do all the voxels afterwards. So I'm not sure how to answer that question, given that I don't really know those methods. But it could be interesting, yes, to compare the two methods.
Yeah, follow up.
Yeah.
Work or analysis could be interesting to compare the two. And it wasn't clear if for your simulation with Inference you do inject noise in the signal, or you do not have noise.
We do not have noise.
So you do not have noise. And did you test what happens if you have noise?
Yes. So it depends on the quantity of noise that you add. So if it's only a small noise, then the summary statistics are slightly off, which makes your posterior distributions to be slightly wider. However, if the noise is too big, then you would also get a bias, and it's wider. So what we try to do concerning the noise is simulate the signal, add the Riccian noise, then extract the summary statistics, and then run the inference with those biased summary statistics. However, we did not get good results. We tried to do like 5 times the addition of the noise given a certain SNR, and then run the inference given. So you have, for 1 theta, you would have 5 times different summary statistics obtained from 5 different noises. But it was helping a bit, having 5 times the addition of noise, but not enough. So maybe we need to do the same thing, but for a lot of more simulations. However, it takes a lot of time computing the signals, extracting the summary statistics, but it's something that we would like to try. Yes.
Okay. Because just as an interesting point, you can add a layer in your network that automatically add noise and can learn the noise. So that could be also an alternative, because classically we add noise on the training set.
Yes.
But there are methods to add noise during training, and the noise level is actually learned by the network as well.
Okay.
So that could be quite interesting to explore.
I agree.
And what SNR are we talking about? You did this test. What is the SNR level?
We tried between 10 and 100.
10
yes. For a fixed value, we did the simulations.
Very much.
Thanks.
And congratulations again. Really great.
Thank you.
Thank you very much. So now we'll just follow my list. So we'll start with Julien Valette asking question, and then we will have Dimitri.
Thank you, Maëliss, for the very nice presentation. I really enjoyed it. I enjoyed also reading the manuscript. I learned a lot about biomechanics. To be honest, that's not my, that's clearly not my part. So I will ask questions mostly in the biophysical model, a little bit in the inverse problem, but not so much. So coming back to the question of Marco about the variable cells. So just to, if you can really explain maybe where you think the contribution of variable cells will be in your model. What compartment would be biased or affected the most by the variable cells?
I think the glial cells will have an impact on the sphere compartment. I'm not sure what site they are, but they would impact the estimated radius or the CS parameter that we obtain at the end. You would get basically an average between the cells, the somas and the glial cells.
So you see there is no fiber-like compartment in the variable cells?
All the fiber compartments would be like mostly like the neurites. However, we did not really get interested into the glial cells, so I'm not sure what how it looks like and what clearly would be the impact.
Okay, because at some point in your manuscript you mentioned that diffusion works show that the variable compartment was the most spherical, which is not what we report actually. Quite the opposite. We think that variable cells, astrocytes in particular, the mostly fibers. And the soma volume fraction of variable cells comparable to normal. So I would expect that the contribution to the fiber compartment is quite large.
Yes, that's something that we should do, and also see maybe even we could add a fourth compartment if it has a different shape than the existing one, something we should investigate.
Now the exchange time is much faster. So I don't. So that's why also it's not obvious what compartment would be both bias. Maybe indeed to consider some kind of extracellular space. There is lots of aquatorial transporters in the bias, so it's very permeable to larger. Something that you cannot take this picture. So that's an open question. I really don't know. There is something that I'm not sure also to understand is the value of student in the soma, either fix or incorporate the CS parameter. I'm a little bit surprised that we didn't try to find some relationship between DN and DS. I don't think they are independent parameters. There might be some relation existing. It's probably not equal because DN incorporates also some relation along fibers, the soma of the nucleus that might affect bias on all these parameters. So they might be not equal, but there might be some fixed relationship between. So we can probably somehow fix the relationship between DS and DN. In case you can estimate the radius of the soma. So you try to do this.
Not at all, because I didn't know whether such relation existed, but we could definitely add it, and it would actually be an extra also summary statistic, which would help us maybe estimate another parameter, an extra one. Or yes, we could use, for example, the results that we have on ATP and GH on trusted regions to try to come up with such relationship during a correlation analysis. That's something that we should investigate in the future.
Yeah. Je sais que the simulation with the neuromorpho approaches you can simulate knowing the actual different coefficients, which is basically the same actually in that case, in the case of the simulation. And then what we extract from the modeling of the data, can have a kind of an effective DS, see how it compares.
Yeah. Yes, we could. The only problem so far with neuromorpho simulations is how do we model the extracellular space and how do we add it, especially if we consider exchanges between the ECS and the other components.
So that's another point, I think. But even without considering the exchange, just the intracellular part, for example, modeling your numerical signal with just a two-compartment model.
Yes.
Like somal fibers. Without the extracellular space, see what you get. And then it will extract some relationship.
Yes, it's something that is doable and we could try that. Yeah.
And so coming back to the extracellular space, can you show your slide, the one with the, again, the
Yes.
So can you explain a little bit more how you define this, what you time practice envelope?
Yes. Well, we basically so the neuromorpho, you have the surface, and we added a space, say around the neurites, and we decided this space, the length of this space, according to which ECS fraction we wanted to have. And yeah.
It's not the same everywhere, like the thickness of the...
Yes, because at some places where you have a bending, then the ECS just overlaps, so it seems bigger. But,
okay. And so what happens when a particle encounters this external, the external side of the ECS?
Yes, I think it's reflected.
So that's probably why you bias. This is not something you suggest, but if you have a restriction in the space, the activation of the longer.
Yes, the model of the ECS is definitely a problem here in the small TX results that we obtain. Yeah.
There is also something you mentioned, it was written in the manuscript. You say that diffusion coefficient was maybe not homogeneous in space. Something I think you mentioned
in the NODI model. I probably mentioned that.
You said that sensitivity is not constant across the whole brain. And you said that it's coming from different microscopy. I was wondering what you understand. The study of metabolites found on different sections of the neurons and extracellular tissue shows that the tissue is not constant.
Yes, what we wanted to say here is that maybe there is, let's say, for example, a Gaussian distribution of the parameters D across the whole brain. However, there could be a difference, though, between two different voxels. And so fixing it directly to one value could be a problem.
But okay, there's no, well, just to be clear, there's no data from spectroscopy showing that.
Okay.
I understand what you
So one question which is probably very naive about that problem. So when you have this low Q-value approximation, high Q-value approximation, if you sample, let's say, more maybe more Q-values, kind of the intermediate range between the low and high Q-value approximations, don't you think that you might increase the precision compared to if you feed directly the signal?
Well, the results that we had showed that it does not increase actually, and that we cannot solve the inverse problem without using the summary statistics.
But that's not considering like a finer sampling of the intermediate Q-value.
Well, in our simulations, for example, using the 10 B values, so between 0 and 10, you have intermediate values, and we fitted the whole signal directly, the average signal of the whole model. In our approximations, indeed, we're only using the large and small Q-value approximations because it's where we can derive those summary statistics, and we don't exactly know how it's what's happening in the middle Q-values. So increasing a lot the p-values is one of the methods that, for example, Olesen et al. Suggest to fit the method, to have a large number, a huge number of q-values with extra large q-values. We are trying to propose something which is more applicable in clinical applications, by restricting ourselves in the Q-value range. Maybe, so also the inversion method that we're using is the standard way to do that. We did not change the hyperparameters. Maybe by adding more transformations, for example, in the normalizing flow, maybe it would actually be able to invert our problem. It's something we haven't investigated because we really wanted something standard and not specially designed for our problem, but we could definitely try that. And maybe those middle range p-values could help.
Thank you. That's all for me. Thanks again. And
Thanks.
congrats.
All right, thank you. So now we'll have Dimitri Novikov asking the questions.
All right. So first of all, great to be here. And the topic of this thesis is extremely timely. The community is shifting from white matter to gray matter. And so you basically describe white matter standard model, and then where does it work? So it was a pleasure to read and to think, because I think all of us basically share this perspective that we're all interested in this, and so it was a pleasure to hop on the plane from New York and come here in person to see all of you guys and be present at your event. Thank you for inviting me. So let me actually be a little bit technical, but I would like to emphasize not technicalities, but physical meaning.
Mm-hmm.
For that, let's go to slide number 20. Great. So let's look. The most interesting term is the q-squared one. Let's forget about q cubed. This is just noise floor. Forget about all the imperfections, right? Just talk about physics, right?
Yes.
So we see that there are terms that when q goes to infinity, they're constant, they're regular, right?
Yes.
And this q-squared term is singular. It grows indefinitely.
Yes.
When q goes to infinity, right? Where does it come from? And what is the nature of this divergence? What is the physical origin of this divergence?
This divergence is coming from the nerds. So this is coming from the in our model, considering that the molecules are going only in one direction, straight in one direction.
Yes. Straight in one direction, meaning that
the radius is considered negligible.
the radius is 0. But how
how does it translate, the zero radius translate to this divergence in Q squared? Indeed, this is actually correct. I just want to make it more physical intuition. So what is the return to origin probability? I mean, when we integrate this propagator over all Q,
yes,
that's actually the probability density to come
back at the original
at the
position.
origin,
Yes.
right?
It's,
Now, of course, this probability density cannot be
yes.
right in real life.
Yeah.
There's no infinity in physics.
Mhm.
Yet in this model, there is an infinity,
Yes.
right? So what is this infinity? I think you're almost answered this question, but just
So the infinity comes from, so the inerts being non-zero.
yes.
So do you mean the diffraction on the membrane of the?
Well, so if you have a stick, right?
Yes.
The diffusion is fine alone,
Yeah.
right? But it's infinitely narrow. So if I look in terms of its shape, it's a delta function in x and y,
Yes.
and something nice in z,
Mhm.
right? So if I ask myself what is the return to origin probability, is the value of the propagator right there. So you're basically saying it's the delta function at zero. Of course, it's an infinity.
Yes.
And that is the infinity
Mhm.
that essentially the power Q squared is because you have in 2 dimensions, an x and y, the zero. Therefore, by just dimensional analysis, you have Q squared divergence. Okay? Okay, good. We understood that.
Mhm.
What is immediate implication of this intuition to every return to origin probability map ever published in literature before?
So
You know, there are papers that are mapping return to origin probability for more than 10 years, right? In say white math.
Mhm.
Do these maps represent the tissue properties or something else? Is it a well-defined metric?
No, then no,
No.
it doesn't, because it doesn't reflect actually what's happening, because it's diverging towards infinity.
Exactly. So it means that it's a function of my protocol much more than the property of the tissue,
Mhm.
right?
Yeah.
I can in fact modify the protocol just to get you any predetermined value about TOP. I'm not studying the tissue at all.
Okay.
It's not a criticism of your work. It's just putting it in
Sure.
protocol.
Yeah.
Okay, great. So that's about other people's work. Now about your work. So let's just still understand a little bit better where this Q squared actually comes from.
Mhm.
So can you sketch the signal from this neurites from the Callaghan model as a function of Q? From what part of the signal does this Q squared come from? From small Q or from large Q?
From large Q.
Right. But so at large Q, it does behave as 1 over Q, right?
Yes.
But this 1 over Q cannot persist to 0. Q equals to 0.
Yes.
So how does the propagator of the signal behave as a function of Q? And you can just take a marker and draw.
Yeah, sure. But,
At Q equals to 0, it must be regular, right? The diffusion signal at Q equals to 0 is normalized to 1.
yes, yes, got the probability, but
Right.
exactly, yes.
Yes.
But then I guess I'm not sure I understand the question.
Sketch the behavior of the signal that you integrate.
The whole signal.
The signal from the stick,
Yes.
right? Before you take the integral over here, the actual signal,
Okay.
the orientation average.
The actual signal is the power law, so it's for the.
That's the power law for large.
Yes, for the Q.
But I want to
Before.
sketch the whole function as a function. It will have direct implications to what follows. That's it's not just a Gedanken exercise. It's important.
Yeah.
So you know basically that at q equals to 0 it's 1.
Yes.
At large q it's 1 over q.
Yes.
So just take a mark and draw it. Signal is a function of yeah.
So if the Q
Yes.
and the signal, so start at 1.
Yes.
So I guess it's something like that. I don't know how it would.
Yes. So around 1, the cumulative expansion works, right?
Yes.
So it should be 1 minus something times q squared.
Yeah.
And then q to the 4th, etc. Right?
Mm-hmm.
And of course, then at some point it transitions to 1 over q.
Yes.
Right?
Yes.
Okay, great. And at which q does this transition happen? Parametric. I mean, there are not many parameters in this model. You basically have time and
yeah.
So there's nothing. You can basically get one parameter out of dimensions of q.
So this would happen when the movement is bigger, so that we can negligible radius. You can negotiate it.
So there is a diffusion length dt.
Yes.
And that's the only parameter, square root of dt, that has this length scale, and q is the inverse length.
Yes.
There's nothing else in this problem.
Yes.
Okay. So this transition from inverted parabola and 1 over q happens at q of the order of 1 over square root of dt.
Yes.
Yes. So this is important. 1 over square root of dt. Maybe write it right there. It's important in both forms. Okay, great. So you see, this function has very two different behaviors.
Yes.
Now you integrate over the whole q, q squared dq, right?
Yeah.
And of course, from 1 over q, you get this q squared, right? But what happens? What is the contribution of that left part before this critical q?
Before.
Yeah, the from 0 to this 1 over square root of dt. This contribution, where is it? Let's estimate. You can approximate the signal as 1 from between 0 and this critical value, 1 over square root of dt.
Yeah.
And you just take it's in 3 dimensions. So it's just 1 over square root of dt cubed. That's just the volume of this thing.
Right? Yes.
Okay. Can we write it down that it scales as 1 over dt to what, 3 halves?
1 over dt. Sorry, I didn't hear the.
Well, it's 1 over square root of dt cubed, right? Because you find the volume in 3 dimensions, right?
Yes.
Yeah, so it's 3 halves, right? Because the q is 1 over square root of dt, and 1 over square root of dt to the power 3, it's 1. Yeah. Yeah, exactly. Yeah, exactly. Okay,
yes.
great.
Yes.
And that will be multiplied by fn, right? Because it's when you write.
Yeah, exactly.
Okay. Now let's look at this term, and let's look at the second equation in the second term in that equation.
Yes.
You see that there is a functionally absolutely the same contribution. Right? That comes from extracellular space.
Yes.
But there is also the same. This comes from inference.
Yeah.
You see what I mean?
Yes.
That so you were after this growing cube square term, which you calculated correctly. But on the background of this term, you lost the term order 1 that doesn't diverge.
Yes.
But that is of the order of everything else. And in fact, it will inevitably bias.
Yes, my estimation here.
So
yes.
That's my major comment on this. So you may want to evaluate it precisely with all the coefficients, add it, redo estimation, we will see how the parameter values will change.
Yes. Yes. Thank you for the explanation.
Okay.
Yeah.
I mean, should I continue or maybe
we can have another round or
Let's proceed in new questions.
Sorry.
The last question comes at the end.
Okay.
So I'll just follow my list, so I do one, but Elena just go. Yes, if you prefer that first.
All right, thank you. So thank you, Maëliss, for the very nice thesis and presentation. So I very much enjoyed your work. I especially think the introduction part, the bibliography, was extremely clear. So it was very nicely written. It's a good reference actually to. So congratulations for that.
Thanks.
Thank you for also making me part of this jury, Damien, and pleasure to discuss gray matter microstructure because a lot of us here are involved with it, so it's always nice to participate. So a few questions. So some of them have been mentioned, so I'm not going to come back to that. But let's start also because you're in the middle of the diffusion theory, just much simpler than me. Can you explain? Sometimes I felt there was some waffle in your introduction, but can you explain what the differences, for example, is between anisotropy and non-Gaussian diffusion?
Yes, so anisotropic means it's mainly going in one direction. And yes, I think I see your point. Anisotropic, I would represent it as the sum of two compartments, like an exponential of minus d squared, and the addition of two of them. Similarly, it's what in Sun is the standard model.
So can you have, for example, an anisotropic medium that is Gaussian?
It would be a sum of Gaussians in anisotropic. Isn't it?
I mean, if you have a sum of Gaussians, it's not Gaussian. You can, like your extracellular tensor, for example, in the standard model, it's approximated as Gaussian, yes, anisotropic.
Yes,
so you have
it's
different diffusivity in different directions, but in each direction, you assume that your displacement can be approximated
yes.
by a Gaussian function.
Yes.
Okay.
Mm.
Okay. You also mentioned in your introduction that a lot of the problem behind this was that we were limited in the scan time and we were interested in providing something that is clinically feasible. And then I think you said that if we had enough number of B values, say we were not limited by the scan time, essentially we could estimate the model parameters without problems. Do you think that's true?
I guess, yes, using an all model, if we're taking, assuming the physics is correct here, if we take the very high Q values, like even bigger than what we have in HCB, then it would be possible to have less biased summary statistics. However, in clinical range, I guess it's harder to go really to achieve really high Q values. And this is one of the main limitations and something we are trying to avoid. Like we are really trying to make something more clinically feasible.
One thing I thought, I mean, I would have liked to see as a graphical representation. So you calculate all these approximations of the signal, either in the low Q regime and the high Q regime. Did you simulate the full signal because you have analytical formulas, and then actually make sure that your approximation really follows the signal at low Q and high Q as you expect, that it's actually a good asymptotic?
Yes, we did to verify our equations and the models, and we had a pretty good approximations for both approximations.
Sorry, I have to skip. So can you maybe go to your extra slide where you have these spin doctor simulations? I think it's we had these as an extra slide to your presentation. Yeah, these ones. So okay, so I also was wondering if you had added noise for this.
Yeah.
I was a bit confused because your curves generated by the simulator for different exchange times, they are nearly overlapping. I mean, you don't really have sensitivity to this exchange time. In your your signal is, am I reading this wrong? Right, so the solid lines are for an exchange time between 10 and 100 milliseconds, which is 1 order of magnitude, and your signals are nearly identical. So would you really hope to be able to estimate something like the exchange time given your acquisition parameters that you're choosing?
So here, I really think we have a problem in the simulator. So the curves that we obtain are probably biased due to the ECS, notably. So I would expect to have a difference with respect to TX for the simulator. Here we don't get it, but we're working on improving the way we simulate the neurons, the whole signal.
Because I think, I mean, it's highly relevant to try to estimate the exchange time. I really don't see it just as a nuisance, like you have to account for the exchange in order to get the other parameters right. I mean, you have to do that, but I think the exchange time is interesting per se. It can be some kind of proxy for membrane permeability. But then to start with, you need to place yourself in a regime where you can actually, I mean, your signal is actually sensitive to that exchange time.
Yes, exactly. And no, it's something we're trying to do, to have like diagram phases where we can exactly point out in which regime we should study these models so that we can have actual valuable simulations and results.
And so to stay a little bit on the topic of this noise versus no noise, so can you maybe go to your slide 30? Okay, so this is all noiseless, right?
Yes.
So I think it's still interesting or useful to benchmark your technique versus something very simple and perhaps dumb as non-linear squares. Because I would argue that actually in the noiseless case, an LLS can estimate all of this quite reliably. So you may have to try different initializations because you may have multiple minima, right? So, but then you can also do it as a frequency analysis. You try like 1,000 different initializations and you would have 2 peaks of solutions. But then, I mean, depending on their frequency, you know which is the correct one. So I think in a noiseless case, I think even NLLS does a good job actually estimating these parameters. So I think it's always relevant to compare to this method. And perhaps once you add noise, maybe you find that this this inference performs better than the method, but it's still a relevant benchmarking.
Yes, it's a good idea to benchmark this way. Although I think
So where does this width come from?
No, so the accuracy of least squares is really good. The precision is terrible.
Yes, but like in this method, without noise, where does the final?
It's probably just a matter of how we plot it.
You learn some fancy ideas, and if you learn without noise, maybe
maybe also our grid of parameter estimations that we choose at the beginning to train for the inference, it might be too big. We might not have enough points for the peaks to be even sharper.
Yes, you probably are not allowing implementation of your fancy model.
Yeah, That's a good question. I don't know.
And then on these HCP data, so you applied your technique to HCP data, right? That's
Yes.
publicly available. Okay. So there inherently you have noise, right? So how do you assess the performance of your technique on these data, given that in the simulations you didn't really assess the impact of noise? So what did you really expect when applying them to these?
We did some simulations with noise, and so what you observe is that the summary statistics are biased. Each of them is slightly biased, and so at the end you end up with posterior distributions which are wider, but still for low a small noise they are still centered on the correct value. It's just that they are wider.
So would you say this is also what you observed in?
It's our assumption, yes.
And so it's kind of like between you and us, because you showed the average of 31 subjects. How did the maps look like on
Yes, we did. I don't have it here. Generally, it's slightly more unstable per subject. When we did the mean, we only kept the values where you had confidence in those estimations, and we wanted to have at least 5 subjects, I think, to consider that a region was stable. And so yes, the results are less trusted in general per subject, and it's also why we're taking the average so that we can then study it in a more controlled way.
And maybe one last question. So I understand maybe you work more on the 3-compartment permeable model, and the exchange one is a spin-off for the next student. I was curious actually because in our endeavors we really found that to estimate this exchange time we needed multi-Q, multi-T data. Like we really also needed data on multiple diffusion times to get any sort of reliable estimate of the exchange time and the practice diffusion. So you seem to be optimistic that you can use multi-shell single T data, which would be great because actually that's what most of available data is like. So can you comment on that? Why do you think you don't have this limitation?
Well, we just derived the summary statistics and found out it could be maybe possible to estimate it. But we actually tried it on ATP MGH, and I can show you the results. So it's totally preliminary results, but as you see, it's most of the gray matter estimations are masked. So we really don't have a great confidence in our estimations, which comes exactly to your comment where it's really hard to estimate it correctly.
Thank you.
Thank you very much. And so the last batch of questions will come from Derek Jones.
Hey, I'd like to offer 3 bunches of congratulations. But first, how are you doing? How do you feel? How do you feel it's going?
I think it's going great.
So do I. So first, congratulations. Well done so far. Secondly, congratulations to the family and friends that have survived all the equations. It's challenging. And then regarding the thesis, I spotted 60 typos. No, that's a typo. 6. It was extremely well written, and I really enjoyed it. So thank you to you and to Demi for having me along. One of the problems of going last in the question sets is all the good questions have already been asked. So I'm not going to start with anything technical. I'll start with something more general. And that is, in this work, you largely studied healthy, normal brains. If I was to ask you about the big picture, why do we want to study cortical changes in the grey matter? And what do you think would be the killer application of looking for changes in, for example, CS, that's going to make a clinician stand up and say, oh my gosh, we've got to get this on that scan?
The big picture, as I mentioned in the introduction, would be mostly to study neurodegenerative diseases, for example, Alzheimer's, who are losing across time neurons. And so I guess the main objective would be to be able to quantify this change through time, and so maybe to better understand exactly what's happening in vivo. And
So I thought you might say that. So I went to page 21 and you said dementia and cognitive deficits appear to be primarily related to neuronal synaptic connectivity. And my question is, have we got the potential using this technique to look at synaptic connectivity change?
no, we don't.
Okay. So therefore, is it the most, if dementia is primarily a deficit of synaptic connectivity, is this the killer app?
No, but I think I also say neuronal loss and synaptic pathology. So it would be more on the first part than the second one.
Okay. So what is currently the biggest and most useful clinical application of diffusion MRI?
Microstructure estimation in general?
Clinically. So what are clinicians using diffusion MRI for the most?
I guess they are using DTI, for example, to estimate the diffusivity.
Okay, so in what condition would it be most useful for looking at diffusivity?
In pathologies where you have a tumor or maybe.
Okay, so have you gone back to the literature sort of around 1990 where we saw the first observations of diffusion anisotropy was in a cat model, but there was something else that was happening there.
I don't know.
Okay, so it's in ischemia. So when you have a stroke, in fact, you know, it's the most widely used clinical application of diffusion because you see a change in the diffusivity that precedes changes in the classical T1, T2 flare images. You know what happens to the diffusivity?
I guess either it drops or it goes much higher.
One of those two. So you're taking 50-50 guess. If you get it right, you pass your PhD.
It drops.
Hey! Right. So it drops. So there's a classic example of where the diffusivity drops. And this is done in a hospital. So what kind of p-value would we use?
Small p-values.
Okay. So what kind of space are we largely sensitized to?
Oh, sorry, could you repeat?
What kind of space intra-extracellular are we going to be largely sensitized to in that?
Extracellular.
Okay. So DE will drop in the pathology. So that was the first thing I saw and thought when you fixed your DE to say it's a third of. So the most clinically applicable use of diffusion explicitly depends on DE changing.
Derek, why do you know that? The intranurite drops and you're sensitive to both. If at low D value, you cannot discount it. It doesn't come from it. It comes from both. We don't know.
Right. But the diffusivity changes, and with the feeding, you see a change in the
Intra drops.
Intra and extra.
Yeah, but I don't think extra drops more than intra. In fact, maybe intra actually drops more.
But both drop.
Yeah.
And the assumption here is that DE is fixed.
I thought you were saying that's only extra.
No.
No.
Okay, so let's carry on. So I just want to come to some basic MRI. When you talk about just the basics of MRI, you say in slice selection, all the nuclei on this plane are processing at the same frequency. If I was to be hypercritical, I'd say there's potentially two things wrong with that sentence. All the nuclei on this plane are processing at the same frequency. What might be potentially wrong?
It's not all the nuclei first.
And why would that be?
It's only water protons.
Yeah, good.
Yes, and the second problem. I don't know.
Well, if the slice thickness is not infinitesimally thin, then why would everything not be processing at the same frequency?
Do you mean the gradient doesn't allow us to select an exact slice, but it's more wide,
Yeah.
and so the frequency in a slice as it has some.
We're just checking that.
Okay.
Yeah. Okay. So you talked about the problem with degeneracy or indeterminacy. And I think very early on you used the expression single diffusion encoding. What do you think would be the most promising way to extend the acquisition to improve this problem of degeneracy? So if you could have longer on the scanner, is it just to continue using more and more Q values, or are there other ways that you may have heard about in the reading that might help us to improve degeneracy?
No, I guess you could also change the acquisition sequence that you're using. There is the SPGE, for example, which has been used, I know, by Chantal Dax et al., for example, about the dot compartment. So I get all the sequences could give us insight into the microstructure.
Okay.
It's just something we haven't investigated at all, but it's something we should do.
And what about other physics phenomena that occur that we're sensitive to with MR that might help us to resolve degeneracy? So what's the first contrast mechanism we hear about when you talk MRI?
It's a T1 or the
Okay.
T2.
Yeah.
Mhm.
So can you see, have you seen anything in the literature that could help us to resolve degeneracy here in the context of this fitting?
Maybe using the different T2 relaxation times.
Okay.
In addition, for example,
we can't.
Yeah.
Okay, so how would we be sensitive to differences in relaxation time? What could we do with our experiment?
in this case, for example, the different F are signal proportions and not volume proportions. So I guess we could use the different time relaxations to move from signal proportion to volume proportions.
So what acquisition parameter would we check?
The acquisition times.
Could I push you a bit more?
Sure.
I mean, we could lengthen the acquisition time, but there's some basic MR timing parameters that we might change. Give you a multiple choice. It could be the inversion time, the repetition time, or the echo time.
The echo time.
Yeah. So you could do a sort of kind of multi-dimensional experiment and like the work from Verard with Teddy, for example. Okay, so let's sort of fast forward down towards the end of the piece and talk about cognitive studies. You say these microstructure estimations from your method could be used in cognitive studies. Can you talk us through how? How would we use them in cognitive studies? Would it purely be looking for correlation, or can you think of a forward model where we might be able to predict individual differences in cognition based on the measurements that we've done?
So far, we only tried correlation studies. Probably other kind of studies could be used, but I don't actually know. We haven't had the time yet to move to real studies of the cognition with respect to the microstructure measurements we obtain.
And then another section, talk about one of our distinguished guests, Professor Jallais, who argues that that unmyelinated neurites do not need to be a definition of a model, because their contribution is dominated by exchange, while Olison suggests a model E and XX that explicitly incorporates them. Okay. So there you've got two people fighting over this. What experiment would you do to arbitrate between these two accounts?
I don't know. If you try maybe fitting both models onto a tissue and look at the posterior distribution that we get, maybe one would be more degenerate than the other, or maybe more confidence in one than the other. That would be my guess.
I forgot to set go on my Fitbit, so I have no idea how much time, but I guess we're pretty much in time.
5 minutes. Up to 5 minutes, so we can be all done. That's great.
Okay, then I'll ask one more question then. Let's go for something very specific, which was in your proposal, in your work, you improved the fitting by using MapMRI to simulate a p-value of 2,800. But that was extremely close to a data point you already had.
Yes.
And it kind of surprised me. I was kind of thinking, well, you're synthesizing this data point from the data you've already got. It's very close to a real data point. I just wonder if you could just talk through the logic of that.
Yes, so for the large p-value approximation, you need at least 3 p-values, and we only had 2. So we wanted to add an extra point using MapMRI. We're not creating a new data, we're more interpolating. So it's information that we kind of already have. It was mostly just so the method could work and we could extract more the summary statistics for the large p-value approximation. That was the whole goal of the addition of extra points.
Okay. Well, listen, I know that we two wanted to have another round question. Okay, done that longer, so I'll say thank you very much.
Thank you.
Thank you very much.
We can do the round in the bar.
Okay. We'll take it offline. All right, so the tradition is to also have your supervisor say a few words, usually kind words, and not ask too many questions. So Daniel, if you want to say a few words, it's that.
Thanks, Alex, for letting me know that I cannot ask questions. I wasn't right. So first, I want to congratulate Maëliss on surviving a real PhD defense with challenging questions and even mathematical examples, which is no minor feat. Does your family understand English?
Not everyone. No.
Maybe I'll switch to French to. Okay. I'll switch to French. Donc, je vais dire que c'était un plaisir de travailler cette fois avec Maëliss et l'avoir évolué d'un début de doctorante où elle s'est énervée quand elle découvrait qu'il avait des papiers qui n'étaient pas tout à fait corrects et qu'il avait un travail scientifique qui disait que le travail d'avant il était mauvais. Parce que ça allait contre son idée de la science de la perfection. Que c'est quelqu'un d'extrêmement discipliné et ça a été un plaisir de l'avoir évolué d'une personne qui est d'un étudiant à quelqu'un qui maintenant commence à diriger, à prendre un rôle de seniorité dans les groupes. Qu'il a fait un travail excellent dans son sujet qui n'était pas du tout évident et que ça se voit par la qualité du jury et des paroles du jury. Et que certainement, elle va nous manquer dans l'équipe maintenant qu'elle part d'un million 5 mois. I don't know if you got this, but I will also switch it to French. I said it was a beautiful thing to see her starting from a PhD student who was getting absolutely angry when one paper was saying that the previous paper was wrong, because for her science was something where every paper was perfect. And then to see her evolve from a PhD student that was hesitating and reproducing the equations of every single paper onto a role where she's coordinating the other PhD students in a seniority role with a lot of passion and a lot of kindness, sometimes. That she will be missed, and that it's now that she's going to like greener pastures and rainier pastures in Paris. And that's it for me.
Thank you.
Okay, thank you very much. Thank you very much. I worked a bit with you, Maëliss, during these 2 years. You had great fun. I hope you had fun like me. And,
Yeah.
Yes. And so I'll stop here. I will not ask any questions, and I will ask you to leave us the room so we can write the report.
Yes.
And we'll probably ask you to come back. Don't go too far. It should not take long.
Ok.
Je reste sur ce Zoom-ci alors finalement, ou bien je passe sur l'autre?
Non, il vaut mieux passer sur l'autre Zoom parce que celui-ci est sur YouTube. Donc, je veux bien que vous alliez sur l'autre.
Ça va, ben, j'y vais tout de suite. Hein.
Merci.
Bon repos.
Et je vais juste couper le micro.
Ouais.
Maëliss?
Yes.
Je pense que c'est plus en avant, c'est vraiment moi, mais pour les photos de la famille, ça va être c'est plus important.
Bon. So, c'est French. Donc, je vais parler en français, le rapport est rédigé en français, ça m'évitera de faire la traduction approximative en temps réel. Donc Maëliss, tu as présenté ce jour tes travaux de thèse sur l'estimation de la microstructure de la matière grise à l'aide d'IRM de diffusion. Dans une présentation très claire et très bien illustrée, tu as su présenter avec pédagogie et rigueur tes contributions allant de la modélisation à l'inférence statistique. Ta présentation a tout d'abord permis de comprendre les bases de la physique de l'IRM de diffusion et d'appréhender les enjeux de l'estimation de microstructure. Et ensuite, détailler ta première contribution consistant en un modèle mathématique de la microstructure, de la matière grise cérébrale et du signal de diffusion correspondant. Une deuxième contribution majeure, tu as exposé ton travail sur l'inversion de ce modèle non linéaire à l'aide des méthodes d'apprentissage statistique, ne nécessitant pas la calculabilité de la vraisemblance. La présentation a permis d'appréhender la capacité de l'approche à estimer l'incertitude sur l'estimation des paramètres. Enfin, tu as présenté brièvement une troisième contribution portant sur l'inclusion de la perméabilité membranaire dans les modèles et dans leur estimation. Tu as répondu avec assurance et pertinence aux diverses questions portant sur les aspects de modélisation et d'estimation statistique. Le jury a éminemment apprécié la pertinence et la grande qualité de ton travail de thèse. Enfin, le jury a apprécié que tu mettes à disposition un code open source à la communauté. Et pour toutes ces raisons, le jury propose que tu sois docteur, proposé, docteur de l'Université Paris-Saclay. Félicitations. Donc maintenant, c'est à toi de faire un discours.
I would like to thank all the jury members for being here today and taking the time.
Maëliss, can you turn the computer? I really would like to see something.
Nice. I will take care of it. It's okay. Right, so just let me be useful here.
I would like to virtually join you. Voila, that's much better.
Okay, so I wanted to thank all the jury members first for being here, taking the time to attend the defense. I would also like, of course, to thank Damien for these 3 years, because I learned a lot, and you gave me my chance, and I'm really glad I could work with you, and I hope we'll keep working together in the future. I would also like to thank all my collaborators, my team colleagues, and my friends and my family for being here today. And thank you very much.
C'est le moment où j'invite tout le monde à boire un verre.
Exactement. Je propose tout le monde d'avoir une coupe de champagne. Je suis à
Bientôt.
l'extérieur de la salle. There will be champagne served.
Ah, the illegal champagne, quand même.
That's the best
Makes
one.
it so exciting.
Wow.
So, Jean-Philippe, you're allowed.
Then I have to go and drink a glass as well.
All right.
Bravo, Maëliss.
Jean-Philippe.
Well, thank you very much, Jean-Philippe.
Bravo, bien joué.
Merci beaucoup.
Une continuation. Vous faites quoi après?
To be
Ensuite,
one of the first to say congratulations.
thank you, Derek.
Bravo.
Good job.
Oui, ça va beaucoup mieux qu'au début.
I look forward to the time. We should again. Do you want,
On se,
now, you probably might not want to remember
on se reparlera, Maëliss.
photograph with all the committee.
Sorry.
Do you want a photograph?
Oh,
with your
oh.
dad?
Or maybe with
Taking a
champagne.
picture?
Yes.
With the
Yes.
champagne.
Yeah, with the
maybe.
champagne.
That could be great. Yes, good idea. Without mask.
On
Pardon.
reparlera parce que là, on est en direct sur YouTube, je crois. Donc,
Oui, exactement.
on reparlera. Mais mettez-moi dans la photo, mettez-moi dans les mains de quelqu'un, d'Eliana.
D'accord. Alors, je vous emmène avec moi alors.
Ouais, ouais.
Je vais arrêter le Zoom, le streaming.
Ouais.